Welcome to the Material Minds podcast, where we talk to the people exploring new materials for a better world. Today we talk with Lara Teruel, a PhD student who's part of developing a futuristic contact lens technology. Now, what she's developing is so sort of dramatic and out of the box that we started this episode a bit differently. We started it with a skit way in the future at a medical technology exhibition where I'm a curious participant and Lara has a stall where she's showing her product to the world for the first time. So, I'm going to ask her all sorts of difficult questions and we're going to see how this is going to impact society in the future. Let's go ahead. Self-replenishing living contact lens. Goodbye, dry eye. By Lara. I should talk to her.
Hi, welcome. Welcome to our stand.
Yeah, hi there. Lara, is it?
Yes, Lara.
Yeah.
I'm Lara.
You seem to be having some kind of contact lens technology?
We do. We have our living therapeutic contact lenses.
Living?
Yes, it's a living drug-eluting contact lens.
Wait, okay. Contact lenses are not supposed to be living. What does that
Exactly.
mean?
We actually combine the technology of contact lenses with a living component. In our case, our living component are biofactories, which are able to produce certain therapeutic. In our first approach of contact lenses, we actually have a hyaluronic acid eluting contact lens. And these biofactories are able to produce the hyaluronic acid in situ. Do you ever heard about hyaluronic acid?
Yeah. Hyaluronic acid. Once I had a kind of a problem with my eye, like a little bit of, I think it was something fell in my eye. It got all red and burny and stuff. And then the doctor told me to get this.
It's very annoying, right?
Yeah. This cheap €5 whatever eye drop thing.
Yes.
And it said hyaluron or something like that on it. And I dropped it in my eye. Oh, I just felt amazing. Like it's just like it was
Instant
burning.
relief.
It felt like instant relief. So wait, you are having something living in there producing this hyaluronic acid?
Exactly. So basically think about for these eye drops that you bought, in the industry, we need to produce the hyaluronic acid later on formulated inside these eye drops.
Yeah.
But in the case of our contact lenses, the factory, it's already within the contact lens and it's added continuously. So I bet that with the eye drops, you actually had to apply them several times throughout the day to get the relief, right?
Absolutely, yeah. It was like, I think maybe a couple of hours or maybe even an hour that I had the relief.
Exactly.
And then, I had to go for it again. And then that's like super annoying because your eye's already swollen, you got to keep it open, you want to put this drop in there. I mean, it's kind of easy, but still half the time the drop falls out and you don't know if it's really in there. And and then they always say one drop at a time and you're not sure if you got that one drop or a half drop or a part of it. Yeah, anyway.
What if I told you that all of those issues could be solved by using our contact lenses? Basically, what the contact lens is doing is, thanks to these biofactories, you can get the hyaluronic acid continuously delivered and in a controlled way. So you have to forget completely about reapplying the drops because basically by using the contact lens, you can continually get the hyaluronic acid released to your eye. And you don't have to do anything else. Just put the contact lens and after the standard period of 8 hours, which is basically a working day, you will feel your eye exactly the same as when you started using the contact lens in the morning.
Okay, that's hard to believe. Wait a minute. So I've worn contact lenses my whole life, and well, not my whole life, but I don't wear them much because they cause a burning sensation by the end of the day, right?
Yes.
Like it's like it's scratching against my eye.
Yes.
And
I agree. I have experienced the same.
Yeah,
in the
you
past.
too? Yeah, okay.
Yes, that's why I currently use just glasses.
Yeah, okay. Interesting.
But with our new contact lenses that we just got, thankfully in the market.
Okay.
With our new contact lenses, you can forget about that because the release of the hyaluronic acid actually helps to improve the comfort of wearing. And this is because hyaluronic acid can act as a lubricant.
Yeah.
You know that in your eye, the structure of your eye, you actually have a small tear fluid layer on top of your eye, on top of your cornea.
Yeah.
And this is where it keeps your eyes moistured and you don't feel any kind of burning sensation.
Yeah.
Sometimes when you have any kind of infection or other diseases in your eye, could be, for example, dry eye syndrome, then your tear layer is actually affected. And therefore you feel this burning sensation. But in the case of hyaluronic acid, as it is a lubricant, what it does is helps to retain moisture, so it helps to restore this tear layer. Therefore, your eye will feel much more comfortable, and also when you blink, it will not disturb the surface of your eye. So you will not get this burning sensation. And the difference is, as I explained to you, that in the case of our contact lenses, you can get throughout the whole day a continuous release of hyaluronic acid. Therefore, your eye will always feel the same across the day. Whereas in the case of the eye drops, you have to reapply because typically, as you explained, the delivery is ineffective. And also in the case of the regular contact lenses, they might have some other strategies to improve the wearing comfort, but hyaluronic acid is actually a gold standard for lubrication. That's why it will make a true difference.
Yeah, I mean, it feels like the way I did when I put those eye drops in my eye, that would be amazing. But hold on a minute. Before, I mean, it all sounds way too good, right? It all sounds way too good. What's the catch here? So you said biofactories.
Yes.
And what is this little factory made of? How does it keep producing hyaluronic acid and giving it to me?
So you might have heard at some point that nature is the greatest laboratory. And we actually took that concept to improve the technology of the contact lenses. And we use living factories, which are basically microorganisms, bacteria. These bacteria have been engineered in a way that they are able to produce by themselves and getting fed by the energy sources that are already in your eyes, or even in the care solutions that we provide with our contact lenses. And with these, they are able to produce the hyaluronic acid in a continuous way.
Okay, wait, hold on a minute.
Yes.
You just said bacteria.
Yes.
And you said engineered.
Yes.
Right? So you have engineered bacteria in my eye. And I'm thinking of like biofilms all over my eye. I'm thinking of infections.
No. Don't be scared. These bacteria are already present in your eye. So you know that.
Wait, what? They're present in my eye?
Yes. Do you know that your body contains a lot of bacteria?
I mean, like down here in the gut, right?
Exactly. But also in all across your body, you have bacteria that constitute your natural microbiome. And in your eye, you also have bacteria. And this bacteria population actually helps you to prevent that other bacteria can conquer your eye and produce an infection. So they are helpful.
They're good bacteria in my eye.
Bacteria.
Like they're in my eye or where?
They are already in the surface of your eye.
On the surface, okay.
And we took this same bacteria, the exact same strain, we call it. And this is the strain that we just gave it a little bit of push so they are able to produce the hyaluronic acid. They are able to do what we want them to do. And these bacteria, of course, are within the lens. So they are not going to leave the lens.
How do you know that?
They are not going to take contact with your eye.
How do you know they're not getting out of the lens?
This is because of the material properties. So we also engineer the material.
You made it that way.
So they are able to prevent that the bacteria could go away of the lens. Nevertheless, they are quite happy inside, so they are able to just produce the hyaluronic acid. They are very happy inside. And also they receive all the nutrients that they need. Because the material of our contact lens is actually a network. Just think about it. A network with pores, and this porous structure allows some small molecules to go through. So basically water or all the nutrients that the bacteria need to eat so they can produce the hyaluronic acid. But they will not allow big things to go away. For example, a bacteria will be too big, so they are just living inside.
So they're good bacteria which you've modified to produce hyaluronic acid.
Exactly.
And because they're good bacteria, would they also just be doing something good for me over there, like what they would naturally do? I suppose so.
Well, basically, they are just focused on producing the hyaluronic acid.
Okay.
So their main function is just eating and producing hyaluronic acid.
That sounds great. Nice.
Yeah.
And they just use the nutrients like in my tears, I guess.
Yes, they will find their food from the natural composition of your tear fluid.
Okay.
And we can also tune a little bit the solutions that we provide with our contact lenses, so they will help a little bit also to maintain them happy.
I've never heard of anything like this before.
It's actually a new concept.
Wow.
And we hope that you are happy to try them.
Yeah, okay, wait. But you tried this in animals and people or something like, and you know that it's safe, right?
Absolutely safe.
Okay. Wow. That's really something. That's something.
You should definitely try them out.
Yeah, I'm gonna look out for them when they're on the market for sure.
Nice.
Yeah. Lara. Hello, Lara.
Oh,
Hi.
Sorry.
Oh,
Hi.
Hi. Yeah, we just started the podcast. I was just asking you,
Sorry,
you have this new paper that you published. Like you were one of the authors on this.
Yes.
Something about contact lenses, and
We're
then you just froze up.
it.
What's going on?
I was just I was just thinking about it, and where we could get in the future, hopefully.
Okay. Yeah. Okay. That's
very
interesting.
Cool.
Yeah, so you have this paper about a contact lens where you have bacteria inside producing hyaluronic acid. This is I came across the paper, of course. Yeah,
yes.
It's very interesting. Maybe tell me a little bit about what have you actually made? What do you have in that paper, and what do you describe?
So basically, how the paper started. We realized that the current technologies for addressing ocular drug delivery were quite inefficient in different ways. So the first, most of the therapies are actually focused on eye drops, and the problem with eye drops is that they actually provide very low bioavailability of the drug, meaning that you have a very low retention time of the drug in the surface of your eye. And this causes that the user, the patient, needs to reapply the drop several times, while also is not very sustainable, as almost 95% of the drug is actually wasted.
Oh.
And this had been a common experience, because I think everybody have tried at least once eye drops, and it's very annoying, the application and having to reapply throughout the day when you actually have an infection or just other symptoms. And on the other hand, the other approach is to actually have therapeutics included within contact lenses. And the advantage there is that as the contact lenses will stay at the surface of the eye, they actually increase this retention time of the drug. And this allows the drug to have more effective, it's a more effective delivery.
Okay.
Nevertheless, for drug-eluting contact lenses, the approach is to load the drug inside the contact lens, which typically is a soft contact lens, which means that it's made of hydrogel. And so inside the network of the hydrogel, the drug will be loaded. But there are some issues regarding the burst release, which means that most of the drug that has been loaded will be delivered in the first hours. Therefore, throughout the day, at some point, you are not getting the delivery anymore. And also sometimes to really prevent this, or to kind of counterpart this burst release, you need to load quite a high amount of drug. And at some point, this could compromise also the optical properties of the lens, which is not desirable.
Yeah.
So we come up with this new concept of having a living drug-eluting contact lens. And in this case, what it means is that in our contact lenses, we are not loading directly the drug, but the factories that produces the drug.
Okay. Yeah.
And our factories are actually living. So that is our living component. These we call them biofactories, therefore. And these basically are engineered bacteria. The strains that we choose are commensal bacteria. So in our first approach, we use Corynebacterium botanicum, which is actually a model for the I commensal.
Okay.
And this allows us we took this strain, we engineered it for production of hyaluronic acid, particularly. Hyaluronic acid acts as a lubricant. So it really increases the comfort at wearing the lens at some point. And we wanted to prove that. So what we did is to engineer this bacteria, put it inside the contact lens, and we chose a design in which we located these biofactories within a hydrogel ring that stays in the periphery of the lens. Therefore, it's completely outside of the field of view.
Okay.
Also, we were able to prove that these bacteria were living happily inside our hydrogel, and they were completely embedded within the hydrogel. Therefore, they are not escaping or leaving the prototype of our contact lenses. And they were able to produce the hyaluronic acid in a continuous manner. We could even, we observed that it was possible to control and to tune if they will produce or not by incubating these prototypes in media that will contain nutrients, whereas in the media that do not contain nutrients, they were all not able to produce a hyaluronic acid, but they were still alive. And also, the continuous release of the hyaluronic acid finally really helped to decrease the friction coefficient at the surface of the prototype.
Okay, wait, just 1 second there. So you have these bacteria inside the contact lens material, right? As a ring. What is this material that's holding the bacteria in there?
The material that we choose is a PVA, it's polyvinyl alcohol.
Okay.
We actually modify it to introduce certain groups that are vinyl sulfone groups, and this allows us to have a photocross-linkable material, which means that upon the irradiation of light, blue light in particular, we are able to cross-link and form bonds in between these groups.
Okay.
Therefore, the long chains of the polymer can actually structure each other to form a network.
Okay.
And this network is stable over time, and it's able to swell and retain water, forming the hydrogel. And what we also observe is that we could actually tune the mechanical properties of these hydrogels by including also sacrificial polyvinyl alcohol.
Okay.
That it's a water-soluble material.
Yeah.
And we can put it inside this cross-link network in a way that over time it gets released after the hydrogel swells.
Yeah.
And this allows more space inside the network. So the bacteria that we encapsulated within these hydrogels were reacting very differently in their way of growth whenever we change these ratios of the two materials.
Okay.
And this was very important for us because we were able to reach a particular composition, a particular ratio that will favor that the bacteria will grow, but they will also be constrained so they will not overgrow over time. Nonetheless, the number of bacteria that we got in the hydrogel was more than enough to provide sufficient delivery.
Okay, and the network is tight enough that you don't have the bacteria just growing out or like squeezing itself out or whatever?
Exactly. Okay. And we also, to make double sure that that is not the case, we actually use one formulation for our ring, which is a little bit less stiff, so it's more flexible and will allow the bacteria to grow comfortably. And in the lens field of view, we use a formulation that only contains the photocross-linkable material. Therefore, it's stiffer and also provides more confinement.
Okay.
So it's like an extra layer. Of course, the ring is within, basically in a sandwich kind of way, inside the contact lens.
And with all these layers, you still have enough, other things are able to pass through, like
yes, absolutely.
what the bacteria needs to grow.
Absolutely, yes.
Right?
We characterize also the hydrogel's diffusion properties. So basically, we studied how proteins with different molecular weights were able to go through, and how fast. So we observed that for the molecules that are required for the biofactories to function, it was completely permeable. And also for quite big molecules was also permeable without any issues. So the hyaluronic acid that, for example, the bacteria produce, it's kind of a big molecule, and still it's completely able to diffuse out. As well as the sacrificial PVA, which also being a polymer is quite large.
Okay, so it's PVA inside, PVA outside. It's different, PVA. Slightly different formulations.
Exactly.
You have a prototype here, I guess,
yes,
right?
Yes, I brought a little prototype. I can show you. So in this one, I only added a little bit of dye on the ring, so it will be visible, if you can see there. So the ring is located in the periphery. The rest is a completely transparent hydrogel. Basically, we are able to shape it in this way by integrating two molding steps. So in the first one, we mold the ring, and later on, we use the hydrogel precursor to place it on top. And we repeat the molding. And basically, we use molds that have kind of this shape. So two-step mold. And we have one layer that can be...
so this is the internal or the concave?
This is exactly, it's like two parts, and you can basically squeeze them one on top of the other.
And then you take this one, I guess, and squeeze it on top.
Exactly. And then we cross-link. And later on, it's very easy to remove. And this is the way we obtain these prototypes.
Pretty nice.
And it's quite an easy fabrication, actually.
Yeah, it looks pretty simple. So
and this is the way that they are produced in the industry.
This is how they produce.
This is completely compatible with the already established industrial process.
And I assume this is a laboratory prototype.
This is absolutely lab made. Absolutely.
It's hand lab made.
We also have commercial molds available depending on the material. The industry uses different types of molds. An advantage of using PVA, for example, because in contact lenses, most of the market is actually focused on silicone hydrogels. They offer other advantages, but the one advantage regarding the fabrication of the PVA based is basically that it is being water based, it will not stick around the molds, and it's very easy to remove, so molds can be reusable.
Then you said that it's this PVA, and then you are able to sense that the friction coefficient changes. How do you knock do you slide the contact lens across a table and see how far it goes?
Not exactly.
Or what do you do?
For that, we use a special device which is called a rheometer. And basically what it measures is we build up this flat. In this case, it was flat. It was not with the rounded geometry. We are adapting now the methodology to measure and to see if it changes with the geometry. But the point is we use these hydrogel discs in which they will contain, there will be completely empty hydrogel, or they will contain the same strain of the bacteria that do not produce the hyaluronic acid, or the bacteria that produce the hyaluronic acid. And they were incubated at different time points, so we will see how it changes over time. And basically, the hyaluronic acid is produced and released outside of this hydrogel disc or prototype. And we can collect it in the supernatant.
Okay.
So we are incubated in quite an amount of liquid.
Yeah.
And then we collect this liquid. And what we do is to place this hydrogel in our biometer, and which consists of 2 plates. And we place on top a drop or a certain sample of the supernatant of each case. And then we measure how is the resistance to the movement of the plates against this hydrogel.
Okay, so it's a flat plate, just moving around like in a rotational manner around the...
Yes.
Okay, yeah, around the contact lens. Yeah, oscillating. Yeah.
Yes.
Okay, and then you can measure the tiny amount of forces that are caused due to friction.
Exactly.
Yeah, okay.
And what we observe is that for the supernatant that was contained in the hyaluronic acid, over time, the friction coefficient will be reduced considerably comparing to the hydrogels that were completely empty.
Yeah.
And for the case of the hydrogels that contain bacteria, but they are not producing the hyaluronic acid, we actually observed that the friction coefficient was higher.
Oh,
which is very interesting because this means that the non-engineered bacteria are actually producing other stuff, part of their metabolism. And this could cause kind of debris and deposition on the surface.
Okay.
Which is not, of course, desirable.
Yeah.
But in the case of the engineered strain, they are focused on the production of the hyaluronic acid.
Okay.
And this is more than enough to kind of...
So hyaluronic acid as a lubricant probably even is helping to reduce the adhesion of whatever other molecules these bacteria could be producing.
Yes, we also have the hypothesis that they might even have anti-fouling properties due to the continuous flow of the hyaluronic acid outside the gel of the hydrogel.
Okay, so it maintains a positive pressure and keeps pushing it out. Okay.
Yeah.
Fascinating. Yeah, so this is... Now you said this is Corynebacterium glutamicum,
yes,
which is a bacterium that's found in the soil.
Yes.
Right? So, and you said it's a model strain.
It's a model strain, else.
Yeah. Okay. So is it related to a strain in the eye?
Yes. We are actually now conducted. We will conduct also studies on Corynebacterium mastitis, which is the actual eye commensal. Nevertheless, we found quite a lot of literature regarding the use of glutamicum as a model for mastitis.
Oh, nice. Yeah, okay. And it is generally recognized as safe strain.
Exactly.
Which in principle could be compatible with the eye. We don't know yet.
We don't know yet, exactly.
Yeah, and I understood that you also did some biocompatibility tests in
we performed.
Yeah.
Of course, all in vitro studies.
Yeah.
That we analyze against some also. This is not my expertise. We did this in collaboration, but some cell lines were chosen according to the standard tests. And what we observe is that there is not toxicity coming from the material nor the presence of the biofactories.
Okay.
Therefore, the whole construct was
compatible with in vitro tests.
In vitro tests.
Yeah.
We are moving forward, continue to work with this, but the first tests were very promising. And And so you said you did it in collaboration. I also assume many parts. This is quite a multidisciplinary project. Absolutely,
can you, who did you work with on this? Different aspects of it? So
for the cytocompatibility, we actually who performed the test are part of the group of Lara Teruel. So I think it's...
she was a guest on the podcast also.
Yes. And for example, for the characterization of the surface at the friction coefficient measurements, we also got collaboration with the group of interactive surfaces.
Roland.
With Professor Roland.
Yeah, who was also on our podcast.
Yes. Yeah. And they are actually moving forward to extend the measurements to considering the geometry, which... Of course, it's a challenge in practical aspects. But yeah, they are also working on that, and we hope to have further collaborations in the future.
Yeah.
And yeah.
And how about the biofactories, the bacteria?
The biofactories were actually engineered by the group of Professor Whitman.
Yeah,
so this
Christoph
is a group,
Whitman.
Christoph Whitman in the university.
Yeah,
Yes.
okay.
And they work quite a lot with glutamicum. And yes, it was a very, very successful collaboration.
Yeah.
And yeah, we look forward to continue working with them. But we would also like to extend eventually the approach and also consider other strains and continue to work with your group at INM. So that
yeah,
will be
so
for the future
maybe
very
we
interesting.
kind of end on the point of future perspectives. So you said, working with other strains and so on. And
Yes.
hyaluronic acid, for example, is actually a cheap-ish therapeutic
Yes.
that you can get, despite the the annoyance of having to use eye drops regularly and so on. But still, you can get that. Maybe just a statement on the actual therapeutic use of
Yes.
it, and then what other therapies could be developed with this platform.
Yes. So in our case, this was the first approach to the concept, and also to prove that our idea actually worked. But the idea is to extend the approach to particularly thinking about pharmaceuticals, which are very costly, and the approach of typically to produce a biopharmaceutical. I'm not sure, but I imagine that it involves quite a lot of steps from the production, then the isolation, the purification, and the formulation.
Yeah.
Whereas in this case, the biofactory is doing everything at once and inside the lens.
Yeah.
So
Okay.
the approach is much more convenient.
Yeah.
And also considering the sustainability aspect, because in the case of eye drops, it's not compatible for quite costly pharmaceuticals, because you are actually wasting most of the drug.
Yeah.
So it's not really a convincing approach. And in this case, the continuous delivery would be quite advantageous.
Okay.
And the fact of the idea is to extend as a platform in which we could basically have different strains or the same strain but with different engineering that could produce different therapeutics.
Yeah. Okay.
Therefore, we can, in the same type of contact lens, using the same material, we can actually just change or update our biofactories and already have a complete different delivery.
So the current one is lubrication. Is probably the best for dry eye disease.
Yes.
And in the future, you're looking towards therapeutics that could treat, infections, inflammation,
Exactly.
tissue damage,
Yes.
and often
Especially
these are expensive
for-
proteins that you need
Yes.
to treat these diseases.
Absolutely. And also thinking about those treatments that are not really friendly with the patient, like intraocular injections, which is quite awful. Maybe this could be an alternative for that.
Okay, so there is a possible way to get from the surface of the eye into the inner part of the eye.
Exactly.
Okay, yeah, that would be very interesting. to see
That would
how
be
it
very
develops.
interesting. I agree.
Yeah. So
We could also think of combining therapeutics and also the hyaluronic acid release, which will be challenges, of course,
Yeah.
but it could also be an idea, because the lubrication also helps to keep the patient happy.
that's
with
comfortable in
it's
the
for
eye.
user experience.
Yeah,
yes.
absolutely. Yeah. No, that's wonderful. I think there are quite a few perspectives for the future, and it's a space that is worth looking out for.
Yeah, we're very happy about it.
Yeah, wonderful. It was great talking with you, Lara, and
Thank
to
you so
learn
much
more
for inviting
about-
me.
Yeah, absolutely. To learn more about these self-lubricating living contact lenses
Yes.
that you're developing. Good luck to you.
Thank you so much.
And yeah, of course, we also collaborate
Yes.
in the lab. So yeah, good luck to both of us in the future.
Thank you.
And yeah, thanks a lot.
Thank you.
So thanks everyone for listening to this, slightly different type of podcast episode we had this time. It's a very exciting new development that we're bringing up here at INM. You know that we're there on all sorts of social media sites. You know that we're there on YouTube. Hit subscribe, like, all that sort of stuff, and watch out for the next episode. See ya.